So this will be the second portion of the renal physiology notes. We previously discussed the functional anatomy of the nephron and many of the aspects with it. And now we'll pick up a little bit on the physiology related to the nephron. A couple reminders. When we look at the nephron, which is the structural and functional unit of the kidney, it has three primary purposes in terms of forming urine. It does filtration. Remember, filtration occurs at the glomerulus. All right, so here's our glomerulus up here. And it filters plasma from the glomerulus into Bowman's capsule, initially into that area between the visceral layer of Bowman's capsule here and the parietal layer of Bowman's capsule on the outside. That's called Bowman's space. Once it gets filtered, it's called ultrafiltrate. Now, ultrafiltrate is a name we give to the filtered fluid because it's protein and formed element free. So it doesn't have any protein, and it normally doesn't have any red blood cells, white blood cells, or platelets. However, anything else that's dissolved in plasma will be filtered through and become part of the ultrafiltrate. So that's called filtration. Now, as the ultrafiltrate travels through the tubules, remember there were three parts to the tubules: the proximal convoluted tubule, closest to the glomerulus, not shown here because they think it's extraneous detail, would be the loop of Henle, and then the distal convoluted tubule here. So anything that gets filtered and becomes ultrafiltrate, if it does not get reabsorbed, that's what this R going back into the blood where the peritubular capillaries are. That's the second part of the portal system of the kidney. So as the fluid goes through the tubules or any other substance, it needs to be reabsorbed. Otherwise, it'll end up being excreted in the urine. So that reabsorption happens not exclusively, but primarily in the proximal convoluted tubule. It happens through a variety of different mechanisms. It can be active or passive transport. Many times it requires some sort of protein transporter. So reabsorption is primarily concerned with the PCT. Things that get reabsorbed would be water. We filter about 180 liters of fluid a day, which is about 125 mLs per minute. That's the glomerular filtration rate. That's the magic number you should remember in your head, 125 mLs per minute. However, we certainly don't pee that much, which means that the average person has about a half to 5 mLs per minute of urine formation. So if you form urine at a rate, let's make the math easy, of 1 ml per minute, and you're filtering 125 mLs per minute, that means you're going to reabsorb 124 mLs of water per minute. Okay? We also reabsorb glucose, and that happens exclusively in the proximal convoluted tubule. We reabsorb all sorts of ions like sodium, potassium, calcium, chloride, typically, and hydrogen ion as well sometimes, depending on the condition of the body. We reabsorb amino acids. We reabsorb vitamins. We reabsorb bicarbonate. We reabsorb BUN, blood urea nitrogen. We saw that in the calculations for clearance in the laboratory. And those are just, some of the examples of substances that we reabsorb. And again, if we didn't reabsorb it, it would end up being excreted in the urine. So that's what that yellow E is here at the end of this tube. Now, also, as fluid travels through, perhaps we did not filter enough of certain substances into the urine to pee out. So we can also, which is the opposite of reabsorption, do secretion. Now, secretion happens typically in the distal convoluted tubule. It should say DCT right here. I can't change that because it's part of the figure. So it should say DC, distal convoluted tubule, right? And secretion is taking stuff basically out of the body, and in particular out of the capillaries, and putting it into the tubule for excretion to basically pee it out. It depends on the acid-base condition of the body, but as an example, we often secrete hydrogen ion, H+, into the distal convoluted tubule. We secrete things called xenobiotics. Those xeno means foreign, so these are foreign substances like penicillin. We secrete a waste product basically called creatinine. We secrete ammonia. We excrete secrete a substance called PAH, para-aminohippocratic acid. I wouldn't ask you what that stands for, but just PAH. As a matter of fact, classically, you see PAH used for secretion all the time because typically it's 100% secreted in any other waste product we might have. So the net of what happens in terms of the kidneys then, we filter fluid into the kidney that becomes ultrafiltrate. As that ultrafiltrate travels through the tubules, we take the stuff we don't want to pee out of the tubules, reabsorb it back into the body. So that's reabsorption, that's the R. And at the same time, and typically happening later in the tubule, in the distal convoluted tubule, we secrete stuff we want to get rid of in even higher concentration than what's been filtered. And what's left is what we excrete as urine. So that's the three processes we need to form urine: filtration, reabsorption, and secretion. Filtration happens exclusively in the glomerulus to form ultrafiltrate. Reabsorption happens mostly in the proximal convoluted tubule, and secretion happens mostly in the distal convoluted tubule. So now, one of the things that really dictates the filtration rate is the Starling forces that you might have looked at in terms of Lab 21. And the Starling forces are the 4 basic forces that either push fluid out of a capillary or push or pull fluid back into a capillary. Okay, so those some of those forces is called the NFP, or as you see up here, net filtration pressure. And if NFP is a positive number, that means we favor filtration. We favor pushing fluid out of the capillary into the surrounding space. And that, in this case of the kidney, would be our glomerulus filtering fluid. The rate at which that is accomplished is called the glomerular filtration rate, or GFR. So you see GFR down here. The net filtration pressure directly determines GFR. GFR is considered to be the homeostatically regulated variable, but GFR is determined to a large extent by NFP, net filtration pressure. So GFR is proportional to NFP because it's determined by it. So as an example, if NFP went up, GFR would go up. As a matter of fact, we could be even more specific and said, if NFP doubled in a normal person, that would be very, very unlikely and abnormal, but if it did for some reason, if NFP doubled, it went up 2 times, then GFR would also go up 2 times. So it would go from an average of 125 mils per minute in a normal person, and their NFP doubled, then GFR would go up to approximately 250 mils per minute. Likewise, if NFP goes down, GFR goes down. Since GFR is homeostatically regulated and doesn't change much throughout the day or even individual to individual, then the mechanisms that affect NFP also generally don't change much. So if NFP is greater than 0, as a positive number, it favors filtration, and NFP is less than 0, it favors reabsorption. For the glomerulus, you should always have a positive NFP because it should always be pushing fluid out of the glomerulus and into Bowman space to form ultrafiltrate. So basically, again, what happens is the blood comes in the afferent arteriole. It goes through the glomerulus. There are two forces that push or pull fluid out of the glomerulus. The major force is the hydrostatic pressure within the glomerulus, right? So that's the pressure of the fluid within the glomerulus pushing fluid out, okay? The other force that pulls, in this case, pulls fluid out would be what's called the osmotic or oncotic pressure within Bowman's space or Bowman's capsule. Now, when we look at the osmotic pressure refers to the pressure due to the dissolved particles. Particularly, the most important pressure we're interested in is actually called oncotic pressure, much like if you looked at somebody who went to oncology, right, and studied cancer or something like that, then we look at the oncotic pressure. The oncotic pressure is the pressure due to dissolved proteins. And essentially, in a normal person, since the ultrafiltrate should not have any protein in it, that's part of the reason we call it ultrafiltrate, then there should be a zero value for the protein outside the glomerulus in Bowman's capsule. So that's usually zero. You can't totally ignore it, because if someone had a condition where their filtration apparatus, the glomerulus, was damaged and proteins were being filtered through, then that would affect their GFR. So we can't forget about it, although typically in a normal person it's not an influence because there shouldn't be any protein there. That is opposed, those two forces, to the forces that push or pull fluid into the capillary. So you have two forces that do that. The biggest force that happens there is the glomerular oncotic pressure. So again, it's the pressure due to proteins in the glomerulus. The reason why we say pressure due to proteins is because when fluid gets filtered out of the glomerulus into Bowman's space, the dissolved particles that are relatively small, which are most of them, they filter through freely. So the glucose and the sodium and the potassium and other smaller molecules dissolved in the plasma get filtered through, and they get filtered through at an equal rate. So if you had 2% glucose in the plasma, it would get filtered through at 2% glucose in the ultrafiltrate as well. So that's not any different. So the only place where it's different is the amount of protein. And so sometimes they call it oncotic pressure, which is really just a specialized osmotic pressure that's just due to proteins. So the oncotic pressure pulls fluid back into the capillary and helps to move fluid back in. The other force is outside the capillary that pushes back on the fluid, and that's the Bowman's capsule hydrostatic pressure. So that is the pressure within Bowman's capsule that pushes fluid back. That pressure is usually due to fluid, and if the tubules were working fine, then that would be relatively low. It probably wouldn't be much more than 1 or 2 in terms of that. But let's say, for instance, you had some sort of kidney stone, some sort of blockage downstream from the tubules. If you did that, then the fluid would back up, and as the fluid backed up, that would increase the pressure here, and could increase the pressure so much that filtration stops. And if that were to happen, this area of the nephron would actually swell and likely burst, and that's called a renal cyst. We all have some renal cysts. There's other reasons to have it as well, and some are genetic diseases and things like that. But again, I mentioned in the first lecture that the average person has about 1 million nephrons in each kidney. You really only need 1 million for normal function, so you can lose a kidney or equivalent to a kidney. And as long as you have, probably 200,000, you're probably pretty close to normal kidney function, which means you can do that a whole bunch of times and still have normal function, right? So if you destroyed half your nephrons that way, you'd still have normal kidney function. So those are the Starling forces. Now, a couple other things. We'll go to more detail in the Starling forces as well. I mentioned that the kidney will filter 180 liters of fluid per day into the nephron, and this is the magic number to know. The magic number is 125 mils per minute. So we see this number right here at 125 mils per minute. Should memorize it. Since the plasma volume of the body is about 3 liters, the kidney filters the equivalent of the plasma volume about 2 or 3 times an hour, which means that we turn over our blood volume pretty quickly over the entire day. A typical urine excretion rate is about 1.5 liters of fluid a day. If it's about less than 1/2 liter, right, that's the minimum that you need, and that's because you need to secrete waste in your urine. And if you're not having enough fluid, we cannot concentrate the urine to a greater degree than a certain extent, and so we won't be able to get rid of as much waste as we would need to. So the minimum is about 1/2 liter per day, but the normal is about 1 liter to 1.5 liters. And so if you looked at like how many minutes, right, are in a day, it's a little less than 1,500. It's a little over 1,400. So if we made a mil of urine a minute, that would be about close to 1,500 mLs of urine a day, which is about 1.5 liters. The kidney also takes a large amount of energy, especially the reabsorption. I mentioned that reabsorption can be active or passive transport, but a lot of it is active transport, which means we need a large amount of oxygen to supply energy for that. And so at rest, your kidneys get about 20 to 25% of your oxygen. So the average kidney probably gets over 1 liter of blood flow a minute at rest. So you remember cardiac output's only about 5.5 liters. And if it's getting over 1 liter, you're getting, 20, 25% there. So that's kind of the basic physiology you want to look at there. A couple other notes. When we talked about the functional anatomy, there is a area we didn't talk about. I sort of left out on purpose because it's already, I think, confusing enough for some people. And so we're going to bring in this other part. When we looked at the nephron itself, right? Remember the nephron is comprised of the renal corpuscle and then the renal tubules. And there's 2 parts to the renal corpuscle, the glomerulus, which is the red capillaries in there, and then the Bowman's capsule. And there's 2 parts to Bowman's capsule, the visceral layer that covers the glomerulus, and the parietal layer, that's the outside cup to direct fluid. And then for the renal tubules, the first part is called the proximal convoluted tubule, the PCT. The next part is called the loop of Henle, right? The loop of Henle has the descending limb that goes down. The descending limb is permeable only to water. So the only thing that can go across the descending limb wall is water. Solutes are stuck inside there. In the ascending limb, when we go back up and back to the distal convoluted tubule, it's the exact opposite. We are permeable to solutes, so dissolved particles can move across the wall in the ascending limb, but water cannot. And this allows us to differentially fine-tune or move water without having to worry about the obligatory flow of solutes in it, or move solute and worry about what's called solvent drag, where we move water with it. So those are the parts of the loop of Henle, and then we go to the distal convoluted tubule, right? And then eventually the collecting duct. And what we did is we sort of, to make it easier to look at, kind of drew all this out like the way it is. But what actually happens in real life is the distal convoluted tubule actually folds over and crosses the afferent and efferent arteriole. Now, it's hard to see here, and it's a little subtle, but I think if you looked closely, you could tell. I know this bottom one is the afferent arteriole, for a number of reasons, but the big reason to me is, notice how wide it is. So it's got this nice wide diameter, and the efferent has a narrower diameter. And so the afferent does have a wider diameter, and that allows us to bring more fluid into the glomerulus, which then allows more fluid pressure there and allows for a greater amount of filtration. The narrower efferent arteriole also helps to back up blood because it's not as easily to exit through this, so it can't exit as easily. And so blood has a higher pressure within the glomerulus, which makes it have a greater filtration pressure, which then helps to keep GFR high. So the wider diameter arteriole is always going to be the afferent. The narrower one's always going to be the efferent. So what happens is the distal convoluted tubule folds back over and actually physically crosses the afferent and efferent arteriole. So I'm just going to kind of draw it in right now. So see the lines that I'm drawing in going across the afferent arteriole. This right here, this is where the tubule crosses. Now some books will say this is part of the ascending limb of the loop of Henle, and other books say it's part of the distal convoluted tubule. To be honest, it doesn't really matter, but what does matter is what this area looks like. So I'm going to circle it right here, this area right here, okay? And this area is called the JGA, and that stands for Juxtaglomerular apparatus. Juxta means close to, the G stands for glomerular, right? So it's close to the glomerulus, and apparatus just means it's a network of things. So let's take a closer look at that and see what happens in the JGA. So here's my juxtaglomerular apparatus. We can see the name up here, right? So our JGA. And again, it's an area where the afferent and efferent arteriole are crisscrossed by the distal convoluted tubule. The areas in this JGA are specialized for a number of reasons, and there's two areas that we're going to look at and spend a little bit of time on. So the first area is called the juxtaglomerular cells, or sometimes just known as JG cells. So we'll see them up here, juxtaglomerular cells. And if you look, they are right here in the pink. So I'm going to go ahead and kind of draw them in. Notice they sort of line right along the arteriole. There's also some up here, although they don't show it. And they touch the distal convoluted tubule. And so what happens is in the juxtaglomerular cells, there's a couple things that happen. First of all, they're on the afferent arteriole, okay, and they physically touch the tubule. And inside are a number of receptors, and in this case, we're most interested in the mechanoreceptors. Mechanoreceptors refer to receptors that respond to mechanical stimulation, and we've seen those before in class, but in this case, it's probably stretch and pressure where they're most sensitive to. And so basically, they sense the blood pressure in the afferent, and like I said, not shown here, but there's also some in the efferent arteriole. Since they sense the blood pressure, if the blood pressure is low, they can change their diameter independently to increase the blood flow and keep it normal in the glomerulus. Because in order to have a normal homeostatically controlled GFR of 125 mils per minute, we need to have a normal filtration pressure, net filtration pressure, within the glomerulus. So these mechanoreceptors are always sensing the blood pressure, and they can dilate or constrict the afferent and efferent arteriole to increase that. So as an example, let's say right here, this is the diameter of my afferent arteriole. And if pressure was low going through here, and the mechanoreceptors were able to sense that, then what would happen is they would dilate the blood vessel. And as we dilate the blood vessel, remember, we're going to decrease resistance because we increase diameter, and those are, inversely proportional. That means more fluid is going to go through the afferent, which means now I'm going to have more fluid here, and that's going to cause a greater filtration pressure, and that's going to go ahead and increase the GFR back to normal. The other thing I can do is on the efferent side, right? The efferent, remember, is narrower, so I'm going to draw it a little narrow, so it's not as wide here, right? So here's my efferent, so A here, the top, E on the bottom. And what I can do for the efferent is I could actually constrict it, so I could make it more narrowed. And so if I make it more narrowed, that means fluid has a harder time getting out, which means I also increase the blood here. And as I increase the amount of blood here, the pressure goes up, and that again increases the pressure for the fluid. So why do we say then here, and why do the textbook manufacturer only show these on the afferent side? Well, one of the problems that we have in terms of the body would be if we had, and we'll talk about this later, but if, let's say we had epinephrine released, okay? Now, epinephrine is going to cause constriction of the arterioles. And one of the problems would be if we wanted to affect how much blood volume we had, we could do that by releasing epinephrine. Now, if epinephrine was released, epinephrine could go both to the afferent, right? So here's my E for epinephrine, and my efferent. And what it would do is, under normal circumstances, without knowing anything else, it would constrict the afferent and it would constrict the efferent, and they would sort of offset each other. Because if I constricted the afferent, then that would mean there's less fluid here and pressure would go down. But at the same time, if I constrict the efferent, that means less fluid gets out, so this goes up. And basically, you would offset the effect of each other. So what our body has done to change that is it said, okay, I know that this is going to happen. So what we do is in the afferent, we have a lot more receptors. So I'm just going to draw them like this. So the turquoise would be the receptors there, and the turquoise would be the receptors in the efferent. So since we have more receptors in the afferent, it's got a much higher sensitivity. And really, then, it's the effect of the afferent that really determines what happens for the glomerulus. So even though both efferent and afferent have the JGA, it's the afferent that has the most receptors, so it's the afferent that's going to have the biggest effect and really determine what's happening in terms of glomerular filtration. The other big thing about these JG cells is they secrete renin. And again, notice there's only one N in it, and from what I understand, it's really supposed to be pronounced renin, but I don't know anybody who does. I've never had a professor who did it. But don't confuse it with renin. Renin is an enzyme that, with two N's, that curdles milk in mammals. And we actually take that enzyme and form it into a substance called rennet, which then makes cheese, that curdles milk to make cheese. So the importance of renin, we'll see later with a hormone called angiotensin, but helps to regulate the sodium chloride and water balance within the body, and really the sodium balance. So that's the JG cells. The other portion of the juxtaglomerular apparatus is what's called the macula densa. And densa, dense, that's where it comes from. And macula, you might remember from the anatomy of the eye in anatomy, there's an area in your eye called the macula lutea. And that just means yellow spot. So macula means spot. So basically this is a dense spot. And I'll show you on the next slide, but it's a dense spot of cells. So instead of having like a tubule, right, that have normal cells all the way around, so sort of like a spoke. This tubule has normal cells, and then with the macula densa, it's got this really, really, really high density of these cells. So they're clustered together, they're much tighter, they're not as big. So under microscope, you can see this area right there. I'll circle it, is the dense spot of cells. So here's the lumen, right? Here's the lumen. I'll draw in blue. That's the opening where fluid will go through. But the macula densa is this dense area on the arterial that helps to filter blood. Not filter blood, helps to sense blood. And so it's modified tubule epithelium that physically touches the afferent. It's actually not on the afferent, it touches the afferent. It's in the tubules. And these macula densa cells have osmoreceptors. So osmoreceptors, osmo refers to the osmolarity of blood. So these look at what's the osmolarity. We've learned a couple times, we've talked about it this semester, that normal osmolarity of human blood is about 280 milliosmoles. So it measures that. It has chemoreceptors in it. So those chemoreceptors measure the chemical constituents within blood. And then it's got mechanoreceptors. And there is some new evidence that some of the mechanoreceptors, while we know there are stretch receptors, we're pretty confident now there's mechanoreceptors that are pressure receptors. So there's baroreceptors in the kidneys. The group at UC Davis figured that out in the 1990s. We're the first group, I think, to demonstrate that mechanoreceptors were in the kidney. And now there's some evidence there might even be some flow receptors, and we're not really exactly sure how they work, but certainly, stretch, pressure, and flow are things that we'd want to look at, both of the efferent and afferent arterial, as well as the tubules. And so what they do is they respond to changes in the tubules and make appropriate changes within the arterioles. So that's the macula densa, which is one part of the juxtaglomerular apparatus. The other part are the juxtaglomerular cells. So let's move and look at it in a little different detail. So this doesn't say anything different than it did before, but what this shows the tubule, which is in direct contact with the afferent and efferent arterial. So here's my afferent at the bottom, here's my afferent at the top, and here's the macula densa, where notice these are much more clustered together. There often is multiple layers of them. And here's my JG cells, these cells that line the edge of the arterial right here. So all these are JG cells. I don't want to do that yet. And so those JG cells are what help to look at the pressure and stretch within the afferent and efferent arterial, and then they can dilate or constrict appropriately. In addition, they secrete the renin, which helps to activate a hormone called angiotensin. So those are the JG cells. The macula densa is this purplish dark area here. Those are modified tubule lumen that physically contact with the arterioles. And again, they have osmoreceptors, chemoreceptors, and mechanoreceptors that look at that tubule lumen and also sense what's in the arterioles and are able to adjust things that way. Both of those things help to form what is called the tubuloglomerular feedback mechanism, or just sometimes tubuloglomerular feedback. We'll actually see this when we talk about regulation of glomerular filtration rate homeostatically. And just like we saw for blood pressure, there'll be an intrinsic and an extrinsic method. And this is one of the major intrinsic mechanisms for regulating glomerular filtration rate. And so the fluid flow through the tubules influences and is influenced by the GFR. And so if there's increased flow through the tubules, it means that we have too much fluid going out potentially. And the macula densa would sense that. It would help the JG cells release renin, which would then activate angiotensin II. Again, we'll talk about that later. And then it will also cause afferent arterial constriction, and that would decrease GFR, which would eventually decrease the amount of fluid going through the lumen, and that would get everything back to normal. So the tubuloglomerular feedback mechanism is an intrinsic mechanism to regulate GFR. So we'll come back to that again when we talk about homeostatic regulation. Now, I think we have enough time to talk about the Starling forces. So glomerular filtration rate is dictated by opposing Starling forces, and I mentioned those before. We kind of looked at some of them. Here's a little more detail for each one. And the net filtration pressure, or NFP, determines GFR, as we said before. The NFP is the difference between the forces that act out of the capillary and the forces that act in the capillary, into the capillary. So the out part, the filtration, is the GHP, that's the glomerular hydrostatic pressure. That's primarily due to the blood pressure. Let's say that's 45 in the capillary. The other portion that, in this case, pulls fluid out of the capillary is what's called the COP, the capsular osmotic pressure. Again, you could say oncotic. Since proteins are not filtered, right, they're left in the capillary, then that should be zero because there should be no proteins in Bowman space. The forces that act into the capillary, that's called reabsorption, are the capsular hydrostatic pressure. That's the pressure of the capsule here outside, pushing back. And then the glomerular osmotic pressure, that's the pressure due to proteins inside the capillary that pulls fluid back in. So you can see the arrows here. The COP is small because it's close to zero. The GOP should be fairly large because that's the big force. The CHP is usually pretty small because most of the fluid that gets into Bowman space either gets reabsorbed or goes out as ultrafiltrate. And then the GHP is the largest, and you kind of see it's got the largest arrow because that has the biggest pressure. So if we assign just, to make math easy, the CHP is 5 millimeters of mercury and the GOP is 25. Then we have this NFP equation. This is the exact same equation we had in lab. The forces out minus the forces in. Now we call them a little bit different. So instead of the interstitial fluid, osmotic, or hydrostatic pressure, right, we have the capsular, so we use a C. And instead of the capillary one, we use the glomerulus because it's particular capillary, but it's still the same idea. So we get this formula GHP plus COP minus CHP plus GOP in parentheses, right? So that's the forces out minus the forces in. So now we can replace the numbers from up here. The 45 comes down. Here's my 45. The 0 comes down. That's 0. So replace those. And then we subtract out the CHP and GOP. So we get 45 minus 30. And that gives me a positive 15. So that tells me my net filtration pressure is positive 15 millimeters of mercury. Since it's positive, it favors filtration, and that's what's going to determine our GFR. And remember, that's what keeps our GFR consistent. Now, not everybody has the same net filtration pressure because it depends on a number of factors, but normal net filtration pressures in humans are about 10 to 20. However, my net filtration pressure in my kidney might be 20 and give me a GFR close to 125, while yours might be 10 and give you a similar GFR number because of the anatomy of the kidney and other factors. So the NFP, has a, fairly small range. And again, if my NFP went to 10, even though yours is 10 and it gives you a normal GFR, that would make my GFR go in half or go to what, 62.5. So once you have a normal NFP, that determines your normal GFR. And again, that range could be anywhere from 10 to 20 for most people. Now, the glomerular filtration rate, or GFR, okay, is the rate at which you form ultrafiltrate. Okay, its units are mLs per minute, and it's really the rate at which fluid is forced out of the capillary into Bowman space, and it stays in Bowman space. It's determined by a number of different factors. The largest factor is the net filtration pressure, but also the surface area and membrane permeability matter. So the bigger surface area, the greater filtration you'd have. And so that gets to, if you had half the number of nephrons that were functional, then you'd probably have a higher NFP because you would have half the surface area for filtration, theoretically. So in order to make up for that loss of surface area, you would increase the filtration pressure. And the membrane permeability, how permeable is your membrane? While we all have, very permeable glomeruluses, we do have differences in that as well, and that can be changed through drugs and other chemicals within the human body. So this arrow, right, is showing how much blood, the renal blood flow, eventually gets to the afferent arteriole, and here's the blood flow to every other organs. And we said normally the kidney gets about 20 to 25% of our normal blood flow at rest. So I kind of explained this, but we'll look at the graphic on it. The capillary pressure is determined locally by the afferent and efferent arterioles. And again, I like this drawing because you can physically see the afferent arteriole is wider, the efferent arteriole is narrower. It was not, just a misjudgment of the artist. They drew it on purpose because the afferent does have a larger diameter. And again, that's a decrease in the resistance, which then increases the fluid into the glomerulus, which then increases the net filtration pressure to keep GFR high. So if we were to increase the resistance of the afferent arteriole, constrict it, that would decrease the amount of blood flow going to the glomerulus. Right? Now, you've only got 5 liters of blood in your body, right? Give or take. That's an average blood volume. And let's say, to make math easy, 1 liter was going to the kidney. Now, if I increase the resistance to my afferent arteriole by decreasing the diameter, that means I'm going to have less fluid going and being potentially filtered through the glomerulus. So that effectively increases my blood volume to other organs because less is going to the kidney, more is going to other organs. In addition, that's going to decrease GFR because I have less fluid here to be filtered. And what that's going to essentially do then that's going to retain water. So when we talked about the baroreceptor reflex, we said one of the things that happens as a compensation to a low blood pressure, right, is to increase the blood volume. And this is the way it does it. We also increase the sympathetic stimulation. We increase the amount of epinephrine, and that constricts the afferent arteriole. And by constricting the afferent arteriole, that decreases filtration, which also decreases GFR. That effectively increases the blood volume that's circulating, and that helps to bring blood pressure back up to normal. So that helps to keep NFP where we need it. Now, if we constrict the efferent arteriole, that means less fluid will get out of the glomerulus, which means since there's more fluid here, that's going to increase the hydrostatic pressure. That's, remember, the filter forcing out or filtration, so that's going to increase GFR. So if I constrict the afferent, I decrease GFR. If I constrict the efferent, like here, I increase GFR. Now, another factor to consider is the blood pressure itself. Now, when we talk about blood pressure, we'll refer to something called the regulated range. And basically what the regulated range tells us is this is the range of blood pressure that our body can use and still keep GFR normal. So I cannot find a figure that is what I've learned and what I understand being correct in terms of the numbers, so I'm going to cross these out. And basically, what the regulated range is, it's from 50 to 150. And so I'll look right here, so it's down here at the bottom right. So as long as your mean arterial pressure stays within this regulated range from 50 to 150, right? This regulated range, notice it's pretty flat. There's no change in GFR. But once we drop below a blood pressure, mean arterial pressure of 50, which would mean somebody's blood pressure would be like 70 over 40. I think most of you realize that's pretty low, right? Then we would lose the ability to auto-regulate, and we would start to see a decrease in GFR here. Okay, so we'd see a decrease in GFR. And once we got to a mean arterial pressure, probably 40, a little under, GFR would go down to 0 because there's not enough filtration pressure to push fluid out of the glomerulus. On the other end, right, if you had a blood pressure of what, 170 over 140, that would give you a mean arterial pressure about 150. We can regulate up to about that point. And once we start getting above 150, we can no longer auto-regulate and GFR will increase. But as long as you're between the regulated range, 50 and 150, then changes in mean arterial pressure are not going to affect NFP because we have the ability to auto-regulate. So that's pretty cool in terms of what our kidney can do. So how do these variables affect GFR? And we'll kind of look at some of them individually and see how that can affect things. So the first variable we'll look at is GHP, glomerular hydrostatic pressure. And again, this is the pressure within the glomerulus. It is largely a reflection of mean arterial pressure, except for the fact that we can auto-regulate it. And typically, as long as you're within the regulated range of 50 to 150, hyper or hypotension are not going to affect it. Okay, but once you get outside that regulated range, that can happen. That can also, which will be affect blood pressure as well. You know, severe dehydration can cause problems here with the hydrostatic pressure. Problems with the narrowing of the blood vessels within the kidney can also cause some issues there. But the big one is really related to blood pressure. And again, that regulated range allows us to keep GHP pretty constant. COP is the capsular osmotic pressure. And the capsular osmotic pressure, again, is the pressure due to proteins. Since normally proteins shouldn't be there, there's not much of an effect of it, as I said before. So that would be proteins sitting out here in the capsular space. They normally shouldn't get through the wall. However, hypertension, as an example, actually helps to push proteins through those filtration slits. Remember, the podocytes have the little feet, the pedicels, and where they intertwine, there's a filtration slit. And we can push fluid and proteins through those filtration slits if we have a high enough pressure inside. And that's what hypertension will do. And that eventually, you know, helps to destroy the filtration apparatus. So once we start getting proteins here in the capsular space, that's going to pull fluid out. So normally, You know, we're not going to see it because it's not there. But if we do and we start pulling fluid out, that's going to favor filtration because it's just going to suck fluid out. And again, once we get proteins within the Bowman space, there's no mechanism to bring them back into the body because they're not supposed to be there. So you will pee them out. And actually, one of the hallmarks of acute hypertension that most clinicians don't recognize is polyuria. People increase pee because of the proteins that get filtered through. Going on the other side, right? So this is the putting fluid back in, the reabsorption side down here, CHP and GOP. The capsular hydrostatic pressure, right, is the pressure of water basically pushing fluid back onto the glomerulus. One of the big places where we'll see that are occlusions in the tubule drainage, like kidney stones. The normal kidney stone actually doesn't develop until you get to the renal papillae at the end of the collecting ducts in the renal sinus, in the the minor calyces and the major calyces in that area. But if you were to clog up one of the tubules by closing off the collecting duct, often with a calcium deposit, most kidney stones start with what they call a nidus, a center of calcium. That would back up fluid and push back on the glomerulus, and the higher this went from the lack of drainage or the impaired drainage, the higher this is going to be, the less filtration we're going to have. And then finally we have GOP, that's the glomerular osmotic pressure, and that's the pressure and proteins that are in the glomerulus. And a number of things could affect that, especially liver damage. If you don't have a functional liver, you're not going to make a whole bunch of plasma proteins because that's where they're made. Protein malnutrition as well. You need the raw materials to make them. And so somebody who's malnourished, they wouldn't have a big GOP, and that would change GFR. And really kind of look at it is, you should memorize this equation. So you should, for lecture, be able to write this equation out and figure out, hey, what would happen if COP went up, right? Everything else stayed the same, so COP goes up. So I come over here and look, and it's okay, I know this equation, so I'm going to go COP is going to increase, right? Everything else stays the same. So since COP goes up, that means the forces, these are the out forces on this side, are bigger than the forces in. And since this is bigger, and since the COP is going to get bigger, that's going to make the difference bigger, NFP will increase. All right, I'm running out of memory because it's not doing it. So if NFP increases, that means GFR is going to increase appreciably as well. So that's kind of how you look at those. And we'll do some group problems on those as well, probably. So this seems like a good place to stop this video, and we'll pick up the next one in the regulation of glomerular filtration rate homeostatically.